Welcome back to the deep dive. Today we are really cracking the code on how our immune system talks to itself. When you think about immunity fighting off cancer or dealing with autoimmunity, it really boils down to cells communicating, coordinating things.
Exactly, and that communication, it's run by these specialized proteins called cytokines. They're
Yeah.
basically the language, the essential language of the immune system. They direct traffic, start inflammation, signal stand down. Really powerful stuff, which is why, they're such huge targets for new drugs.
Right, but for, well, decades, we've only had bits and pieces of this language, like trying to translate a whole language with just a few phrases.
Yeah.
We never had that global map. We didn't know how every immune cell type responds to every cytokine signal all at once.
Yeah, it's like trying to understand, say, the entire internet just by watching one single phone line. You miss the big picture, the context. And that's the massive problem the study tackled. They basically built what they called the Immune Dictionary. It's this super comprehensive collection of single-cell profiles. Think of it less like a list, more like a detailed translation guide. It shows who gets which message and exactly how that message changes them.
And the scale here is just staggering. That's where the real power lies, right?
Hmm.
They looked at responses to 86 different cytokines, pretty much all the major ones we know,
Across more than 17 different types of immune cells.
which adds up to over 1,400 unique combinations of cytokine and cell type.
Yeah.
It's a lot of data points.
And crucially, they didn't just do this in a dish. They did it in vivo, inside a living mouse, specifically in the lymph nodes.
The immune system's headquarters, basically.
Right, where all this communication naturally happens. And the final data set, it included, get this, 386,703 individual cells.
Wow. Okay, so our mission today is to really dig into this dictionary. What does it tell us about specificity, like who's listening to what signal, and also about how flexible, how plastic these immune cells can be. And then we need to look at this tool they built, IREA, which lets researchers use the dictionary on real-world data.
Yeah, from cancer treatments to, severe COVID-19 cases. It's pretty exciting stuff.
Okay, let's unpack the methodology first, because building something called an immune dictionary sounds like you need to be incredibly careful. Why in vivo? Why lymph nodes specifically?
Well, lymph nodes are critical. That's where your main fighters, the T cells and B cells, hang out and get activated. Doing it in vivo is key because cells don't act in isolation. They're in their natural neighborhood, bumping into other cells, getting other signals. Context is everything. The final response isn't just about cytokine A hitting receptor B.
Makes sense. And the protocol itself sounds very precise. They injected 86 different cytokines one by one under the skin near the lymph nodes.
That's right, under the flank skin, which drains right into those inguinal lymph nodes. But the timing, that was really critical too.
How so?
Well, if they waited too long, say 24 hours, they wouldn't see the initial message anymore. They'd just see the downstream effects, the chaos that follows. So they collected the lymph nodes just 4 hours after the injection.
4 hours. That's really quick.
Yeah, it's an early time point. Early enough to catch the cell's primary transcriptomic response, basically,
Yeah.
which genes turn on or off right after getting that signal, before everything gets complicated by secondary waves of immune responses.
And the breadth of cytokines they covered. That wasn't just the famous ones you mentioned, IL-1, common gamma chain, IL-6, IL-12, IL-10, IL-17, interferons, all types, TNF, complement growth factors,
Pretty much the whole landscape. If it's a known immune messenger, they likely profiled it.
and the cell types too, not just B cells and T cells and NK cells,
Right.
but all the dendritic cell flavors, PDC, CDC1, CDC2, macrophages, monocytes, even those rare stromal cells like FRCs, over 20 types in total.
Right, a really comprehensive view of the whole immune ecosystem in that node.
So what did the raw data first show? When they mapped out all those 386,000 cells, did they group by the cytokine they just saw?
No, and that's really interesting. The primary grouping, the biggest organizing principle, was the cell's inherent type. A T cell still looked like a T cell, a B cell like a B cell. The signal, at least at 4 hours, didn't fundamentally rewrite their core identity.
Okay, so identity is king, but they could still see the effect of the cytokines.
Oh, absolutely. Within each cell type cluster, the stimulated cells clearly separated from the control cells that just got saline. And when they looked at the genes changing the differentially expressed genes, or DEGs, They found an average of about 51 DEGs change per cytokine cell combo, though that varied hugely, from 0 changes to over 1,500 genes in some cases.
And mostly activation, right? You said 72% were upregulated.
Yeah, the general theme was turning things on. And they double-checked this with known examples. Like, you give IFN beta, you expect SG15 to go up. And sure enough, it did, robustly.
Okay, so we have this incredibly detailed, validated map. Gene X changes in cell type Y 4 hours after seeing cytokine Z. What's the first big surprise or major finding from looking across all this data?
Well, the first big story comes from looking at it cytokine by cytokine. And the overwhelming theme was specificity alongside pleiotropy.
Okay, break those down. Pleiotropy means one cytokine can affect lots of different cell types, right?
Exactly. But the key finding here was how it affects them. It's not sending the same message to everyone. When one cytokine hits multiple cell types, it generally gives each one highly specific, tailored instructions.
So it's not like a broadcast announcement saying everyone panic. It's more like a general alert. But then each department, T cells, macrophages, B cells, gets its own specific checklist of things to do based on that alert.
That's a great analogy. Take IL-1 beta, a major inflammatory cytokine. The dictionary showed it triggers a distinct gene program in almost every single cell type it interacts with. It's like it's conducting an orchestra, telling each section exactly what part to play to create this coordinated inflammatory response.
But were there exceptions? Any cytokines that did act more like a universal alarm?
Yes, the type I interferons, IFN alpha-1 and IFN beta. These are your classic antiviral signals. And they acted much more uniformly. They induced common antiviral gene programs, basically sets of antiviral genes across almost all cell types. It's like the universal intruder alert for viruses. Everyone runs the same basic defense drill, mostly regardless of their specific job.
Okay, that makes sense. Now, here's where it gets really interesting for, say, developing drugs. You mentioned secondary effects.
Right. This is a huge point. When you give a patient a cytokine as therapy, maybe to boost cancer-killing NK cells, you might think you're just talking to the NK cells.
Yeah.
But this map revealed how quickly things spiral.
How so?
Well, they looked at potent T cell and NK cell activators, things like IL-2, IL-12, IL-15, IL-18. These are used or explored in cancer therapy. The dictionary showed that these cytokines hit the NK cells, yes, but almost immediately induce those NK cells to pump out huge amounts of IFing. That gene encodes interferon gamma, another really powerful cytokine.
Wait, so the NK cell gets the go-kill signal, but its first response is to shout another different signal to everyone else.
Precisely. That interferon gamma then blasts out and strongly stimulates B cells, dendritic cells, macrophages, creates these strong secondary effects in cells you didn't directly target. This could explain a lot of the side effects we see with cytokine therapies: unexpected inflammation, toxicity. You start one fire, but it quickly triggers 3 or 4 others.
Wow, okay, that's a massive takeaway for anyone designing these therapies. You inject one thing, but you might be activating a whole cascade almost instantly. You have to account for that crosstalk.
Absolutely. It highlights the complexity. Now let's flip the perspective. Instead of focusing on the signal, let's focus on the cell receiving it. The second major discovery was all about cell plasticity. How much can these immune cells change their state, their function? We call this polarization.
Right, the classic example is macrophages. M1 for fighting, M2 for fixing. But this study looked beyond macrophages.
Way beyond. Because they had single-cell data for everything, they could systematically map these functional shifts, these polarization states, across all the immune cell types.
And what did they find? Is polarization common?
It's basically universal. They identified more than 66 distinct cytokine-driven polarization states across the immune system. It's not just a macrophage thing, it's a general property of immune cells.
Okay, let's start with macrophages then. Did it just confirm M1, M2, or was there more nuance?
It definitely added nuance and complexity. IFN gamma, that secondary signal we just talked about, pushed macrophages into the MACB state. That's your classic M1-like pro-inflammatory fighter, making things like 6CL9 and 6CL10 to recruit other fighters. But IL-4 and IL-13 induced a different state, MAC. That's more M2-like, the healer repair type, marked by genes like retinal and Grill ACAX. So yes, M1, M2 is real, but it's driven by specific signals and likely exists on a spectrum.
Got it. Now, you mentioned a really novel finding with natural killer cells, the NK cells. We know they kill things, but IL-18 did something totally unexpected.
Yes, this was fascinating. They found this NKF state. Now, cytokines like IL-2, IL-12, IL-15, they induce the expected cytotoxic state, making NK cells better killers, turning up the G's amine gene. But IL-18, It induced this NK state that upregulated over 1,000 genes.
1,000 genes. That's an order of magnitude more than most other cytokines, you said?
Exactly. It wasn't just tweaking the killing machinery, it was like a massive system overhaul.
So what makes NKF so different? What were those 1,000 genes doing?
That's the key. It was polyfunctional, highly diverse functions all ramping up at once. Yes, there was cytotoxicity, DZMB, but also huge increases in biosynthetic processes, suggesting the cell was gearing up for growth and division, driven by genes like MyClec. It was also turning into a recruiter, making XCL1 to call in dendritic cells, and it was upregulating master control switches like BATF that handle cell differentiation.
So IL-18 doesn't just make the NK cell a better assassin, it turns it into, like a field commander, coordinating recruiting, growing the response.
That's a really good way to put it. It suggests this IL-18 to NK cell axis plays a much broader, more strategic role than just direct killing, a command center function.
That's incredible. Did they see similar plasticity in other, maybe less studied cell types?
Yes, they found examples everywhere they looked.
Yeah.
B cells could be polarized by IL-4 into an IL-4I1 positive state. Even gamma delta T cells, which are kind of unconventional T cells, could be pushed by IL-23 into a TGDF state that cranked out IL-22, a cytokine important for tissue repair. It just reinforces that polarization, this ability to change function based on signals, is a fundamental feature across the board.
Okay, so putting this all together, the specific responses, the plasticity, the researchers could then map out the whole communication network, right? The cell-cell interactome. Who talks to whom?
Exactly. They could chart which cells produce which cytokines and which cells respond to them based on the dictionary data.
And did anything jump out about the structure of that network? Like, who were the big communicators?
Yes, something really striking. They found this inverse correlation. The rarer an immune cell type was in the lymph node, the more distinct types of cytokines it tended to produce.
Wait, really? So the less common cells are actually the biggest talkers, sending out the widest variety of signals?
Kind of counterintuitive, right? But yeah, think of them as specialized communication hubs. The real standouts were fibroblastic reticular cells, FRCs.
Okay, what are those again?
They're stromal cells, part of the physical structure of the lymph node, not typically thought of as frontline immune cells, but they express the highest number of distinct cytokines. Based on the dictionary, these rare FRCs could potentially influence almost every other immune cell type in the node.
Wow. So don't ignore the background players. What about more traditional immune cells?
CDC1's a specific type of dendritic cell, known for presenting antigens to killer T cells, also emerged as major hubs. They used IL-1 beta, that inflammatory director we talked about, to influence a huge range of other cells. It suggests that if you want to modulate the whole immune response, targeting these rare but highly connected hub cells might be really effective.
Okay, this is all fascinating biology, but how do we actually use this dictionary? It's massive. That's where the software comes in, IREA.
Exactly. IREA stands for Immune Response Enrichment Analysis. It's basically the practical application of the translator tool, because just knowing a cell has a receptor for a cytokine doesn't guarantee it will respond or tell you how it will respond. Receptor levels can be misleading.
Right.
So IREA uses the actual response signatures, the patterns of up and down regulated genes they mapped in the dictionary to look at gene expression data from, say, a patient sample and infer which cytokines are actively driving cell states and polarization in that real-world context.
Okay, let's see it in action. They tested it on cancer immunotherapy data, specifically single-cell data from tumors after patients got anti-PD-1 checkpoint blockade therapy. What did IREA find?
It painted a perfect picture of what you'd hope to see in a successful response. IREA inferred that macrophages inside the tumor were being polarized towards that MACB state, the M1-like pro-inflammatory anti-tumor state driven by IFN gamma. It also inferred that NK cells were shifting to the NKE state, that's the cytotoxic killer state induced by IL-2, IL-12, and IL-15. These are exactly the kinds of activities you want that therapy to trigger.
So it confirmed the known mechanisms of action. Did it also pick up on things that might be hindering the therapy?
Yes. It found that the strongest negative enrichment, meaning the signal that seemed to be actively suppressed or counteracted in the treated tumors, was for TGF-beta 1. And TGF-beta is famously immunosuppressive, known to help tumors evade the immune system. So IREA correctly identified both the pro-LSL and anti-LSL tumor signals active in that environment.
Very cool. And they also applied it to severe COVID-19, which we know involves a massive cytokine storm.
Yeah, and again, it worked beautifully. The IREA analysis of patient blood cells immediately highlighted strong response signatures to specific cytokines in B cells and T cells, and these inferred active cytokines lined up perfectly with the cytokines known to be dramatically elevated in the blood of severely ill COVID-19 patients, the ones driving lymphocyte dysregulation. It allows researchers to sort of deconvolute that cytokine chaos.
Okay, so wrapping our heads around this, the Immune Dictionary isn't just a catalog. It's revealed much deeper complexity, way more specificity, and frankly, surprising plasticity across the immune system. Polarization isn't niche, it's everywhere. And even one signal, like IL-1 beta, sets off this incredibly coordinated multi-cell response.
Absolutely. It gives us a molecular framework, a map, to go beyond just guessing based on receptors. We can now use transcriptomic data to deduce which communication lines are truly active, which cytokines are driving things in almost any disease. It's a huge step forward for rationally designing therapies.
All right, so here's the big thought to leave you with. This incredible dictionary was built using a single, pretty high dose of each cytokine, given one at a time, and measured at just one early time point, 4 hours. Now, if one isolated signal can trigger such a complex, cascading, network-wide response with secondary effects popping up almost immediately, imagine what's happening in a real patient getting, say, combination immunotherapy. Multiple cytokines potentially hitting at different times, different doses, interacting in ways we couldn't predict before.
That really is the challenge, isn't it? Now that we have this incredibly detailed dictionary showing just how sensitive and interconnected and plastic the system is, how do we use this knowledge to design the next generation of therapies, especially combinations, that cleverly harness this plasticity?
Wow.
How do we guide the response precisely without accidentally triggering massive, unpredictable side effects from all that secondary signaling and crosstalk we now know is happening?
That's the million-dollar question for immunology and drug development moving forward. How to speak the immune language precisely, not just shout into the storm.